Welcome to this Bright Talk webinar brought to you by MilliporeSigma, the life science business of Merck KGaA, Darmstadt, Germany, and home of Sigma Aldrich and Aldrich Chemicals. My name is Brianna Upton, and I am the Biomedical Materials Product Manager for Sigma Aldrich Material Science. Today's webinar discusses nano and microfabricated hydrogels for regenerative engineering. Our speaker today is Professor Ali Khademhosseini from the University of California, Los Angeles. Professor Khademhosseini received his PhD in bioengineering from MIT in 2005 and is currently a professor of bioengineering and chemical and biomolecular engineering at UCLA Henry Samueli School of Engineering and Applied Science, and is a professor of radiological sciences at the UCLA David Geffen School of Medicine. Dr. Khademhosseini also serves as a director of the Center for Minimally Invasive Therapeutics and as an associate director for the California Nanosystems Institute at UCLA. Dr. Khademhosseini is globally recognized as a leader in combining micro and nano engineering approaches with advanced biomaterials for regenerative medicine applications. He has authored over 500 journal papers with over 33,000 citations and 60 books and chapters. In addition, he has delivered over 300 invited and keynote lectures. Dr. Khademhosseini's interdisciplinary research has been recognized by over 40 major national and international awards, including the Presidential Early Career Award for Scientists and Engineers, which is the highest honor given by the U.S. government for early career investigators. Professor Khademhosseini is also a fellow of the American Institute of Medical and Biological Engineering, Biomedical Engineering Society, the Royal Society of Chemistry, Fellow of Biomaterial Sciences and Engineering, and the American Association for the Advancement of Science. The Sigma Aldrich catalog includes an extensive selection of biomaterials for tissue engineering, including gelatin methacrylate. We work diligently to continue to expand our portfolio with innovative biomaterials for tissue engineering and 3D printing applications. Please continue to bother us with product suggestions for what we should be adding to our catalog offering. I would also like to take this opportunity to highlight the technical resources about biomaterials for tissue engineering and 3D printing for free viewing and download under the Attachments and Link tab. With this, I'd like to hand over the floor to Professor Khademhosseini for his presentation.
Thank you, Brianna. I want to start off by thanking Sigma for this great opportunity to present our research and the wonderful collaborations that we've had over the years. For this talk, I thought that I will highlight a few different things that actually represent some of the work that we're doing, future directions, as well as some of the collaborations that have come out from our work with Sigma. And just to let you know a little bit about the lab's overall picture, we're very much interested in making materials, particularly hydrogel materials, that can be used to control the behavior of cells and make tissues. So because we're interested in controlling the architecture of these tissues, we often make our hydrogels in a way that they can be cross-linked with light, temperature, pH, interpenetrating networks, and adding different types of nanomaterials to them to make these hybrid systems. And this allows us to control a range of their chemical, biological, mechanical, and even electrical properties. Now, once we have the right material, as you can see at the bottom here, we are interested in processing this material into different architectures, which is going to be very important for us in a range of different applications. And specifically in the middle here, you see that we're interested in things like 3D printing and bioprinting, particularly, which is spawned off our collaborations with Sigma. I'm going to talk in this presentation some aspects about our development of new materials for medical applications, and then actually spend the last half of the talk talking about some applications on organs on a chip and developing drug discovery systems, as well as some 3D bioprinting techniques that we've developed here. So broadly speaking, our lab is interested in a range of medical applications. These include things like making materials that are used very quickly by clinicians, and that could be things that used for cardiovascular therapy, surgical applications. But we're also interested in making stem cell therapeutic systems, 3D printing, tissue engineering applications, and organs on a chip. So I'm going to just highlight 2-3 stories here today that really show what we've done in the past couple years and shows the ways in which biomaterials can be used in some of these medical applications. So the first story I want to talk is a story that kind of shows our approach to solving medical problems. And this is a story that shows how medical problem can lead to a development of a material and how interactions between clinicians and engineers like myself can actually push some of these things forward. So this is an approach in which we try to develop a minimally invasive STB, which stands for shear thinning biomaterial, for endovascular embolization. Now, what endovascular embolization means is that actually blocking blood vessels. And this story recently came out in Science Translational Medicine, and it's basically a material that is made from gelatin or gelatin methacrylate, and it's combined with a nanoparticle, these silicate nanoplatelets, to be able to make a material that can be used for this medical application. Now, the story actually is interesting because I spend many years at hospitals directly interacting with clinicians, and I was lucky enough to interact with a clinician who's shown here, Rami Oklu, who introduced me to one of his main clinical problems, which is trying to treat blood vessels. And in the picture in the top left hand, you see this is a picture of an aneurysm, which is weakening of the blood vessel that allows bulging and sometimes even this thing bursts, causes internal bleeding. Now, what people like Rami do is that they go inside these blood vessels and deploy metallic coils, which are shown on the bottom left hand here. Now, these metallic coils are supposed to go inside these blood vessels, block the blood vessel by slowing down the flow, allowing this thrombosis to happen inside the blood vessel. Now, there are major problems with these coils because in the bottom right-hand side, you see that this is an example where there's lots of coils have been placed inside the blood vessel, but it hasn't really worked. So sometimes you need to put many of these coils, each one costs thousands of dollars. So this is a very good, and this slide shows a very good example of what I'm talking about. So in this case, this patient has internal bleeding, and my colleague here has put a lot of these coils, as shown in this black region here inside this image. And what he has done afterwards is tried to see if these coils have blocked the blood vessel. And he put some contrast, as shown in the middle top image here. And it basically, what you see is that this contrast, which shows in the black dye, goes right through the blood vessel. And you can see in the right-hand side that basically the coils have not been able to stop the bleeding. Now, if you actually look inside the GI tract, you see that the bleeding here would continue. And these kinds of patients could have a lot of major issues, potentially even having to have major surgeries or even dying. So what we did is that we came up with this material. This is a material that tries to address this problem by kind of behaving like a toothpaste. It's shear thinning material. As I mentioned, it's made from gelatin and nanoclay, and it's a family of materials because this combination of these two different materials allows us to make a range of different materials that have different ratio of these two different components, or the total amount of the solid component in this gel is going to be different. So just to show you how this behaves in a mechanical way, you can look at the bottom right-hand image here that basically shows that the material can be delivered through a needle or a catheter, but once it comes out, it actually maintains its shape. So it is, in many ways, you can think about it like a toothpaste. Now, if you want to actually look at this material and look at its mechanical properties, you can put it inside an Instron machine and play around with it. Here you can see two graphs that shows the behavior of this material. The one on the left is what is called yield stress, or amount of force you need to make this material become liquid-like. And what you see here is that three different concentrations, and as you change the amount of these nanoplatelets in these materials, you see that the material is very different. So you can get materials that are very strong, as shown by the 9% red line here, or materials that are very weak, as shown by the black line here. Now, the material is also reversible. So when you apply the force, it becomes liquid-like, and when you remove the force, it goes back to its original mechanical properties, which is what you can see here on the right-hand side, which with various formulations here, that when you apply the force, the mechanical properties go down significantly, and then they can be recovered. Now, with this particular material, the reason why we made this particular formulation is that the material is made from intrinsically thrombotic material. So when you put this in contact with blood, it actually forms a blood clot. So here you see these basically circular things here, which are basically bottom of these plates. And when you put the blood in these plates, typically if you just have a dish, it takes 6-7 minutes for blood to coagulate. Now, when you have our material, this is the material compositions are on the left-hand side, is a 9% material that goes from 0% nanoclay, so 100% gelatin, to basically 100% nanoclay. And what you see here is that the amount of time it takes to actually induce the blood coagulation here significantly decreases. So as you can see in basically in the red being present in less than 1 minute, or actually the times that are shown in the graphs on the right hand, very quickly you can get blood to coagulate. Now, why this is interesting is that when you put this material inside the blood vessel, not only it occludes the blood vessel, but also it induces coagulation, so it blocks the blood vessel very nicely. Now, to test any material in a clinical setting, you want to first start simple, see the biocompatibility. These are some studies in which we perform these kind of biocompatibility studies in these rodent models. The row on top shows our material and shows its basically biocompatibility after 3 and 28 days. What you see here is that basically there's a typical biocompatibility initially that basically maintains its behavior over the 28 days. The material slowly degrades and is filled with these infiltrating fibrotic tissue. Now, this is a much better response for what we want compared to other types of material. So this quick clot is a normal material used to stop bleeding right now, and you see that there's a lot of inflammation and there's persistent inflammation even after 4 weeks. Now, the next thing you want to do, if you're comfortable with the material's biocompatibility, is to put it inside these blood vessels. And we started off with simple blood vessels of rodents, and we put the blood, in this case, the material in the femoral artery. On the left-hand side, you can see that this is the femoral artery, and we actually ligated it, put the material inside, and we opened the blood vessel. And on the right-hand side, on top, you can see where the material is with this green that's shown on the top of the skeleton here. And when you actually look at the blood flow, normally you would have flow on the left leg here, but the left leg flow is completely stopped because you actually put this material inside the femoral artery, so it blocks that. Now, this is good for treating a rodent, but how would it actually work in larger blood vessels? To do that, we then do the next thing, which is prepare ex vivo experiments. Here, we can actually take blood vessels, in this case from pigs, and see how well this material is designed. So here we take, in the middle, you can see the pig blood vessel. What we do here is that we put the material inside this blood vessel and to occlude it, and then we pressurize behind it. We put a pump and a pressurized system so that we see that how much pressure you need to dislodge the material. And one of the things that you see here is that on the right hand, when you look at the amount of pressure required, basically you need pressure that's many times the venous pressure and multiple times the arterial pressure to dislodge the material. So if you put the material inside these blood vessels, it stays there and is not dislodged under physiological conditions. So then Rami and I got excited and we said, can we actually try to do this in large animal models? This is actually my friend performing these in these large pigs as a preclinical model to actually testing this in humans. And what we see, we've done a lot of experiments here. I just show a couple slides here that shows how well this works. This is one example. The arrow on the left-hand side shows the basically common iliac artery, which is one of the bigger blood vessels in the body. And what you see here is that the blood vessel initially is open, as shown on top here. But once you put the material inside, as shown in the bottom picture and the right-hand side picture, the material occludes or stops the blood vessel. And on the right-hand side, you can see the zoomed-up image here where you can see the tip of the catheter and the top arrow, as well as the bottom arrow, which shows the edge of the material, showing that basically this has blocked the blood vessel. Now, there's questions that you want to ask about how well this works. One way to do it is to actually ask the question, can parts of this actually get dislodged and go downstream and cause strokes or heart attacks or things that you don't want? Well, one way to do that is to actually put the material and then see whether pieces of it actually come off. And this is one example where we can actually take the pig and put the material upstream from the lung, as shown in the top middle image here, where the arrow is. We can put the material there. And as shown in the right-hand side, we can then do imaging of the entire lung. We can basically see whether the entire lung's vasculature is open, and we do see that in fact this remains open, so then none of the material that we put in top has actually been broken off and gone inside the lung and caused blockage where you don't want it. And then the other thing that you want to do is to test long-term viability here of this material. So here you can see some of the data showing that what happens after 4 weeks inside these blood vessels. So here, this is the cross-section of the material inside a blood vessel on the top left-hand side. You can see the blood vessel remains occluded. And the image on the right-hand side is zoomed image showing that basically the material, which is shown in this pink in the middle here, is being degraded by the cells that infiltrated from the sides. And what happens is that you basically get a full thrombotic effect, which is basically leads to fibrotic tissue, which is kind of shows it's a permanent occlusion, which is what we want and which is what happens with these coils long term. So this is great results, preclinical model. Now we're trying to actually figure out how to push this to clinical experiments and actually hopefully down the road use it in patients. Now this shows one area, and this shows how we can come up with a medical problem, design the material, and then work with a team, including clinicians and engineers and material scientists, to actually be able to find a treatment for it and try to push it as far as possible before doing clinical trials. Now I want to shift gears a little bit and talk about another area of the work that our lab is doing, which is related to coming up with systems for addressing challenges in drug discovery. Now, this is drug discovery, as you know, is a big problem. There's a lot of companies, drug companies, that try to make and identify new drugs, and this is a big process that takes a lot of time and is extremely expensive. Right now, it's about a decade and over $2 billion to come up with a new drug. Now, the reason these drug candidates fail is, as you can see in the top left-hand image, the causes of failure is mostly due to efficacy and safety of these compounds. So that means that we can't predict whether they're going to work or whether they're going to be safe. Now, the reason for that is that when you actually do these experiments, whether you typically use things that you see in the middle here, static dishes, like tissue culture dishes, or small rodents, which really do not replicate the complexity of human physiology. So over the past few years, there's a whole attempt at trying to recreate these human on a chip systems, which is basically recreating different tissues from human cells. These could be specific tissues for metabolic states or particular mechanical or other kinds of behaviors that the tissue does. And once we have these tissues made individually, we can even link them together in a way that mimics the circulation between these different tissues in our own body to recreate more of a physiological feature here. Now, there's a lot of progress that has been made in this field. So this slide shows some of the different versions of the tissues that are made using these systems. So you can see that blood vessel, muscle, bone marrow, and many other tissue types have been recreated using these organ-on-a-chip systems. And really the goal of this is to take us past this 2D monolayers that use animal cells and our individual cell or tissue types to things that mimic human physiology better, which include 3D organ systems that have primary cells or even diseased cells, and has these fluidic systems that can route the fluid between these different tissues. So that's the goal. Now, I'm going to tell you about our contributions to this. We've actually worked for the past few years with our colleagues at Wake Forest University to actually develop a fully integrated, fully automated system of these human on a chips. And this is an actually image of how these things look. Now, the pink device here at the middle is basically an incubator, which is a small incubator. All our tissues and fluidics are housed inside this system. And all the surrounding system here is actually the systems that we need to monitor what's happening here, to be able to sense what the cells are doing, as well as to be able to control what the fluids are going to wind up doing, where the fluids are going to go, what's going to happen. So if you want to actually zoom in to that pink box in the middle, what you see is that we've actually made these things to be in a modular format. So in the top left hand, you see a schematic of how this thing would look. In the middle box here, this is a breadboard or a fluidic controller. What we do to this fluidic controller is that we can put in different modules, attach different modules that could have different tissue types. It can have different modules to control the fluid flow, like bubble traps and pumps and things. And you can have different modules to control to kind of sense what's happening. These could be physical sensors to look at things like temperature, pH, or biochemical sensors to look at things like what happens to things that the cells secrete. And the rest of the images here shows actually the real device. In the bottom, you see, in the bottom left-hand side, you see what the actual schematic that is on the top left-hand side actually looks like. And then the ones on the right actually are showing the fluidic component here. Now, if I could show you some videos, you could see how controlled these fluidic systems are. You can open and close valves in a computer-controlled way and be able to actually monitor things in real time. Now, once you have the right fluidic system, you want to make the right tissues. And what we do with the tissues is that we actually use these little small reactors that we can open up and be able to put the cells and the tissues, and we often 3D print our tissues directly inside these little chambers. So on the left, you see some of these examples of how these things actually look. In the middle column, you see some examples of the liver modules that we're making. So these are hepatocytes that can be printed either as spheroids or organoids, or we can actually put these cells and then be able to pattern them. And on the right, you can see some examples of the heart tissues that we can make, which is, again, we can make it using organoids or be able to make it using other patterning approaches. Now, when you actually want to make these, it's important to make the right tissue and analyze how well it functions. So this is an example of some of our liver organoids. So what you can see on the left-hand side image here is that we can take these liver organoids and actually 3D print them inside these reactors. So here you can see these basically spheroids are made from many liver cells, and we can 3D print them inside. And we actually use some of the products that are now available by being sold by Sigma, like gelatin metacriloid, to actually house these cells and put these aggregates inside them. And when you actually start looking at different behaviors of these liver markers, they behave fairly well. They express the right kind of markers. And when you look at their expression of alpha-1 antitrypsin, albumin, or ceruloplasmin at the bottom three graphs here, you see that over 30 days culture, they work very well. Now, we've done these kinds of experiments both with liver cell lines as well as primary liver cells from humans. So the next slide is actually going to show how primary liver cells look. So in this case, we take primary cells primary hepatocytes, we mix them with other types of support cells like stellate cells, Kupler cells, endothelial cells in the right ratio. And then what we do is that we actually make these organoids that have all the right features of these liver markers. So the top right-hand panel shows a range of different markers that are associated with a good liver functionality, and we see those expressed. Now, one of the things that you want, again, is to get good viability. In the bottom left hand, you can see that you have very good viability over time. And what's more important is that you want these liver tissues to be physiological in their behavior to real compounds. So there's a couple different drugs on the bottom that you can see here that we've exposed these liver organoids to. And these drugs have been actually recalled by the FDA due to liver toxicity. So they were approved and then recalled due to liver toxicity. And we actually can see the liver toxicity here. So you see green when there is no exposure to these compounds, and you see red, which means the cells are dying when the concentration of these compounds gets higher in the presence of these organoids. So our liver organoids can actually predict and see how these things behave. Now, we also do the same thing with heart organoids. So in this case, we want to make, again, tissues made from human heart cells. So we take induced pluripotent stem cells, differentiated to different heart phenotypes, and then combine them together to be able to make these organoids that are mimicking heart behavior. So the panel on top left shows some markers associated with heart cell behavior and some viability. And you see that basically these are expressed. We can actually look at these cells and look at their spontaneous beating in real time. And again, we see that these things beat in real time as shown on the right-hand side and can form kind of structures that mimic heart cell behavior. And more importantly, when we challenge these little heart organoids with drugs that have been recalled by the FDA due to heart toxicity, as shown on the bottom here, we see that, again, on the left-hand side, you see these organoids are initially green. That means they're alive. And as you start adding these drugs, you start seeing cell death, as shown in the red here. So this is also shown in the bottom right-hand side here, which is beating rate of these heart organoids, again, exposed to different drugs. And what you see here is that they initially beat at low beating rates, but once the drug is at a higher concentration, they do not beat anymore. So this is great. So I've been talking about individual organs so far, but what about actually being able to make larger and more physiological systems? So the way to do that is actually be able to now link these organoids together so that it mimics human physiology that has many different tissue types interacting with each other. So the schematic in the top left-hand side here shows what I'm talking about. This is basically a platform that has a heart module connected to a liver module. Now, why do we want to do that? Because this allows us to have a functional tissue that is very important, like the heart, connected with a metabolic tissue that, again, like the liver, that does a lot of things in the body. So now this heart-liver module can actually start behaving more like a human physiology would. Now, this is a simple experiment that I'm going to explain here. It's what happens if you just take these things and add acetaminophen to it. Acetaminophen, as many of you guys know, affects the liver and causes toxicity. So in the bottom left-hand side here, you can see the heart behavior as shown in things like CKMB expression and relative beating. And what this shows is that as you increase the acetaminophen concentration in this system, the heart does not change its behavior, which is predicted. Now, on the right-hand side here, what you see is the liver behavior now, as you add this acetaminophen to the system. So what you see is that in the images, as you increase the concentration and increase the time, you start seeing more liver toxicity. And also when you look at the bottom right-hand side here, if you look at albumin expression or GST alpha expression, you see that there are changes as you start adding these compounds. So the liver starts behaving differently, whereas the heart does not. Now, this is a simple experiment. It shows that you can have multiple tissues and start probing the system. Now, wouldn't it be nice if you actually see interactions between these different tissues? And this next experiment, I think, demonstrates this interaction better. So in this case, we have the liver and the heart again, as shown in the schematic in the top right-hand side. And what happens is that if we go to the top left-hand side, we start adding a compound, epinephrine. Now, epinephrine is a compound that affects the heart cells and makes them beat faster. So as you add more and more epinephrine, the beating rates of these heart cells increases. Now, this is expected result, and the reason this happens is that epinephrine interacts with a receptor in these heart cells that makes them beat faster. Now, this receptor also has an antagonist, a competitive binder to it. So on the plot that is on the top middle, you see what happens when this antagonist is added to the system. So the beating rate of the heart cells decreases because this competitive binder works in inhibiting the effect of epinephrine. Now, this is what would happen if you just have the heart cells. But when you have the liver cells, as shown on the bottom left-hand side here, the liver cell actually takes this antagonist and makes it metabolizes it and makes it an inactive form. So in this case, when you have, as you're going to see in the bottom right-hand side, when you have just the normal heart cells, you get some amount of beating, which is in the baseline here. When you add the epinephrine, the beating rate increases as expected. When you have this antagonist in the next bar, the beating rate decreases. And when you have the liver cells, what happens is that it starts recapturing the effect of the epinephrine because it metabolizes this inhibitor. Now, this is the kind of stuff that I think is going to really enable drug discovery process in the future and be able to do these kinds of things in real human cells. So then there's another area that this is going to enable is the ability to do even things related to cancer. So here in the top left-hand side, you see another example of what I'm talking about. In this case, we have normal heart tissue, and connected to it, we have a tumor tissue. So the idea here is that can we try to test chemotherapeutic on these tissues and then be able to see at which kind of chemotherapeutic levels we start seeing effects that is going to be bad for the normal tissue. And you want to basically, obviously, affect the cancer tissue, but not the heart tissue. So what you see with a lot of the plots in the bottom left-hand side is basically our ability to monitor what is happening in real time. And so we can basically monitor temperature, pH, oxygen, and have these mini microscopes at the bottom left-hand side that can monitor the entire system in real time. And on the right-hand side, what you see is that we can actually now start seeing what happens to, in this case, the liver as we start adding the chemotherapeutic on the top side. So the liver, you see that as we add the chemotherapeutic, doxorubicin, the liver markers start changing. Albumin and GSD alpha start acting. But at the bottom, we can actually monitor the cardiotoxic effect. So as we start adding these chemotherapeutics, we can see how much it affects the healthy, normal tissue. And these are the kind of experiments that right now are at their infancy, but you can imagine down the road, you can have systems that maybe even patient-derived cells from their tumor, and then you can optimize drug formulations to these tumors outside the body. So I'm gonna, the last part of the talk is just telling you about some of our 3D printing developments. And what I've talked about so far with organs on a chip, you can quickly appreciate that the ability to have the right cells and the right architecture is important. So many different tissue types are basically dependent on the right architecture. Form and function closely follow each other in the body. So what we've been interested in is to recreate the architecture of different tissues, whether they would be some of the ones that I've mentioned here or other ones, using techniques like 3D bioprinting. So what we've done is over the years, we developed a lot of different types of technologies that some of them are shown here. But for today, I'm just going to tell you some of the work that we've done in enabling new types of bioprinting technologies. And of course, the inks are just as important as the devices, and this is why we worked with Sigma in trying to enable the development of some of the inks that are now broadly used. But for today, I'm going to tell you a little bit things about the hardware as well. So we've done a lot of work in actually trying to do extrusion-based bioprinting, and these extrusion-based bioprinters are now, there's many of them that are commercially available, but the general idea is that you can, as you can see in the top right-hand side, have a nozzle. You basically suck your cells and gels in these nozzles, and then you can deposit these things. And on the bottom right-hand side, you can see how these cells and gels, when they're deposited, this is a cross-section of how these things actually look. Now, the reason why this is important is that the architecture of these gels actually starts affecting what the cells wind up doing. So on the left-hand side here, you can see basically an image in which we patterned large pieces of gels and then little fibers of gels. And on the right-hand side, you see how the cells start behaving differently. On the top, you can see that in fibers, the cells start sensing their surrounding fiber architecture. They start elongating in these fibers and forming a tissue that's very organized. Whereas in these larger, just deposited gels, the cells, when they don't sense the architecture, they just grow randomly and make a very dense, disorganized structure. So the ability to 3D print and control the architecture of these tissues is important for making them in the right way that you want. Now, we've done a lot of different development of these. So there's some ways in which you want to print, let's say, blood vessels, where there are ways in which you can actually start printing your agarose structures, in this case, sacrificial material. You can print gels that mimic the shape of what these blood vessels would look like. Then you can pour your cells around it, as you can see in the top left-hand schematic here. And once you have the cells around the printed structures that you can have, then you can actually remove these sacrificial rods and then be able to have something that looks like a blood vessel. So on the right-hand side, you see how some of these blood vessels actually look like. Again, these are done with many different materials, including gelma and other types of systems. But you can see that these start having blood vessel-like architectures. You can have cells around these blood vessel features. And at the bottom, you can see that you can even add endothelial cells and have these endothelial cells line these blood vessel channels. Now, there are ways in which we've been trying to make these inks better and make them more widely usable. So one of the things that many people have done in 3D bioprinting is to use materials like alginate. Now, alginate is good in its printability, but it's not as good in many of the biological needs that one has. So in this particular example, what we've done is that we made an ink that is made from alginate, as well as any other biomaterial combined together. So on the left-hand side here on top, you see that this ink is made from alginate plus some other polymer, for example, gelma. And what we do when we print this is that we print these systems and we cross-link them initially using calcium, which affects the alginate. So we can quickly print these into, let's say, something like a calcium bath, and then the calcium cross-links the alginate network, and then we can go back and cross-link the other polymer network, whatever it may be, and whichever kind of cross-linking method, whether it's heat or light or etc. And then once we have that other polymer network cross-linked, then we can remove the calcium network as shown on the top right-hand side, and then be left with just the secondary polymer network. So at the bottom, you see some examples of this, of having printed both of these systems and then getting rid of the alginate network at the bottom right, at the bottom middle, and then at the bottom right-hand side, you can see the actual scanning electron microscopy image of this. And what we can do with these now is so we can print these things in a very high resolution. So here you can see these coaxial nozzles that are printing alginate, gelma, and cells. And we can, once we print them, we can cross-link the gelma system using light, and then be able to remove the alginate. And we can get fairly high resolution here. In the bottom right-hand side, you see some of our 3D printed structures made from many different layers. And next to it, you can have a coin here. You can see the scale. So the next thing that we do with these, of course, is we have cells inside, and we basically allow the cells to remodel what the tissues wind up doing. So there's a lot of work that we've done in trying to actually put cells, and then over time, as shown on the top here, you see the cells remodel these tissues. And at the bottom, you see some of the examples of these things. So I'm not going to get too much into it, but just to tell you that the goal of these bioprinting systems is to initially recreate some of the architecture, but subsequently be able to allow the cells to do the real tissue engineering. So the other thing that I'm going to talk about is once you print out one of these systems, you realize that tissue architecture is actually requires you to print different cell types and different material types. So current 3D printers sometimes are not fully capable of doing this. So what we have done is that we've taken inspiration from, let's say, a spider and how it generates its silk, and we've been able to take this inspiration and actually use it in 3D printing of many different materials. So on the top here, you can see what a spider does. In the shape that you see of the spider here, you see that in the back, spider has many different glands that it can mix and match in different ways to extrude the silk that it wants. So this is how a spider can generate basically silk at will and control the composition of this. Now, taking this inspiration, we can now start making fluidic channels, as shown in the bottom here, where this is the schematic where you can see many different inlets on the left-hand side here. And each one has a little valve that's computer controlled. And based on which one of these is open, you can now start extruding materials that have different chemical compositions. So taking this inspiration, now you can start extruding materials. And what I've shown here is just an example where in the top left-hand side, you can see that if you have 3 channels, one has this red, the other one has the green, and the other one has the blue dye, and you open each one of these channels sequentially, then you can extrude the material that has these dyes sequentially. Now, if you can open them all at the same time, then all of these will be expressed. So using this kind of tunability, now you can start extruding different materials in different ways. Now, having that ability allows you to do other things as well. So for example, here you can see that in this case, if you have the same 3 channels with 3 different materials, as shown in the top left-hand side here, but you have regions where multiple channels are open instead of, so basically 2-3 materials can come out at the same time, then you don't only change the chemical composition, but you can also change the topography. In the middle, I show you some ways in which you can change the architecture of these on the surface of these hydrogels, or even be able to have multi-phase systems, oil droplets, air bubbles, etc. So what we can do with this now is we can now extrude different cells, different materials at will. So these are some examples that we can extrude these fibers, where on top you can see that different parts of the fiber can have different cell types, and this allows us to build complexity into our system. And at the bottom, what we can see is that we can have, even in these fibers, we can have different regions where different molecules can get released from them, and then be able to have these drugs come out and affect the surrounding biology. So we've now taken these kinds of concepts, attached them to 3D printers, and used our different bio inks to be able to deliver them. So here you can see that this is a simple bioprinter in which there's 2 different inlets, as shown in the schematic on the top left-hand side. And when you open channel A or channel B, you can start extruding different materials. So you can make basically 3D printed structures that are made from the 2 different inks in different layers, or even in the same layer of the 3D printing structure, have both of these materials be present. And now what we're doing with this is now we're pushing this. We made versions of these 3D printers that have many different inlets, and we can mix and combine these different inlets in different ways to be able to get many different outputs. So just as you have all the colors that you see right now come out from the 3 primary colors, this is the same kind of concept that you can have a few different inks and then be able to mix them together in different ways to get the different behaviors that you want. So where are we going with this? So we think that there's a lot of advantages to these kinds of printers. So typical printers, on the left-hand side here, you basically see how as you print, how much delay time you have as you change the material. So, and basically, as you print different materials, you don't have any delay time. So on the right, there are videos of how typically these things would work, where you print with one material, you change the nozzle with a different material. But with our continuous printing systems, we don't have these issues. We can rapidly print different materials. And then what we're doing now is we're generating 3D printed tissue structures from these systems. So here you can see that we're printing different architectures with different types of inks and combining different bioinks with different capabilities to be able to do that. And I think in the future, there's going to be a lot of advantages in trying to enable these kinds of engineered tissues using these systems. So I want to end the talk there and open up for some questions, but I did want to leave you with a couple parting thoughts. One is that I think that right now there's a lot of opportunity in this area of 3D printing and bioinks. So there's applications in making tissues ever expanding. When I first started doing these, people were always just thinking about regenerative medicine as the endpoint. But now we're starting to appreciate that you can study biology, you can make disease models and enable the drug discovery, you can make sensors from these systems, make even robotics and even artificial food using these engineered tissues. And what I've been really impressed with is really the incredible group of people that have worked together to enable this. So I really want to thank them for all the work. We've started in UCLA about 6 months ago and already grown significantly. But without the people who were in Boston, as well as the people at UCLA now, none of this work would have been possible. And of course, great collaborators and the funding sources. And then the last thing I want to talk about is, maybe I'll just put this slide up as we answer question. I think this is a field that is really got a lot of interesting opportunities. I think Sigma is doing a lot of great things in actually enabling this field by the products they're making. But I like this painting of René Magritte. This is a self-portrait of him. He's a painter, a surreal artist at the turn of last century. And what he has shown here is his self-portrait of looking at the egg and drawing the bird that's emerging from it. And I think in many ways, this field and where it's going is just at its infancy. And I think what we need to do as a field is to enable the bird that's going to emerge from it. So with that, I want to thank everyone, and I'm going to spend the next few minutes hopefully having some discussion and answering some questions. Thank you very much.
Fantastic. Thank you, Dr. Khademhosseini, for a wonderful talk.